Thanks very much for the organizers for putting together this meeting. I don't think I've ever been to a meeting that covers the field from such different perspectives and so comprehensively. So probably my talk will echo a lot of what you've already heard this week. So the title of my talk that you have in your booklet is Angstrom-scale rulers to correlate transport and geometry of metallocene molecules on gold. Really, I'll probably just touch on show you some preliminary data on metallocenes at the very end, but the focus of my talk will be on how we correlate conductance and geometry. So how we can characterize the atomic scale features of our junction to correlate them to the performance of the junction. And so I'll be telling you mostly about these kinds of measurements that we've already heard a lot about, where we have a molecule bound between two metal electrodes. But I just want to tell you a little bit about my lab. We are in general interested in this question of how does the nanoscale structure correlate to function in single molecule systems. And so we also use different type of single molecule spectroscopy to try to get at this question. So in particular, we have optical tweezer type systems where we're able to manipulate molecules using optics. So I started at Boston University in 2017, and so this is a view from the Boston University campus looking at the Boston skyline. So here's the river. So if you're familiar with Harvard and MIT, MIT is just across the river right about over here. And this is Commonwealth Avenue where the campus is located. And for those of you who are interested, Fenway Park is right over here, so very convenient. Another way to characterize where we are is to say that whereas Harvard and MIT are on the red line, BU is on the green line. This is the subway, the Green Line subway that runs through the campus. So we are in the Photonics Center at Boston University, and so this is our brand newly renovated lab that we moved into in December, so just under a year ago. So we have a kind of a wet lab area and then a characterization area. This is the optical tweezer being built. This is looking towards the STM brake junction setup, which is over there in the distance. And as you can see, we still have quite a bit of empty space that we have to fill, which is exciting and also a bit daunting. Okay, so why do we kind of rely on these two different types of techniques? So very briefly, we find basically that they're complementary in many ways. So as we've already heard, scanning probe techniques are good at characterizing really the sub-nano regime, so angstrom to nanoscale. But they are quite stiff probes, right? So actually, something that hasn't been mentioned is that when we do these kinds of manipulations, when we pull on single molecules, we apply quite large forces, so nanonewtons of force. And that's okay if we're looking at probing small molecules, but if we want to, for example, probe proteins, which we heard about on Tuesday, and we want to try to perhaps measure charge transport through a single protein filament, well then nanonewton forces might become a problem because we will just denature our protein and we won't be able to probe transport through the system. So that's where optical tweezers might be more applicable. And so what we would love to be able to do is use some of the plasmonic techniques that were mentioned yesterday to try to indirectly probe charge transport through softer or longer polymers using optical tweezers. But I won't tell you about that today. So today I'll focus just on these kinds of STM brake junction measurements. Okay. So the overview of my talk is that I'll tell you some of our initial measurements in the new lab about measuring conductance and using conductance to kind of benchmark the rest of our measurements. So we use conductance to really get at the atomic level geometry of our junctions. I'll tell you a little bit about what we see when we go do temperature-dependent measurements and how we are able to get conductance that seems to scale with temperature. And then at the very end, I'll show you briefly some preliminary work on ferrocene. Okay, so I don't need to go into a lot of background details about transport on the nanoscale. Everyone here is obviously very familiar with this. I just want to say that why am I so focused on the geometry of our single molecule metal junctions? Well, it's because this kind of idealized picture of discrete molecular orbitals bound between this continuum of energy states in the molecule, in the metal, is, of course, changed once you bound the molecule to the metal and you hybridize the spectra, right? And so the way that the molecule binds to the metal will affect how these resonances are broadened, and thus it will affect transport at the Fermi energy. So, as has already been mentioned, all the transport we observe is predominantly this tunneling, so super exchange mediated transport. And why is it so hard to find examples where we have near-resonant tunneling? I would argue it's because of this problem of when you bind the molecule to the junction, you have to immobilize the molecule on your metal. And so in order to do that, you form a chemical bond. And so you transfer charge into the molecular orbitals. And by doing that, you shift them away from Fermi. So we can actually see that if we do STM, if we deposit molecules on the metal, and we look and we find a step edge where we know the molecules are bound more strongly, and then we do IV curves, we see that on the edge where the molecules are bound more strongly, the molecular resonance has shifted away from Fermi. And so these kinds of effects are very important in considering what kind of transport we will get. So again, to emphasize this point, yes, structure of the molecule determines transport, but I would argue that it's actually the structure of the entire junction. So we can't just say conductance of molecule A is something. We have to really characterize what is the geometric features of the entire junction as a whole. In other words, we cannot neglect the interface anymore for these nanoscale systems because the interface is on the same size scale as the molecule itself. Okay, so how do we measure conductance of our molecules? So we have this STM-based break junction. It's a very simple setup. So I was very excited to hear about all the really interesting developments using spectroscopy that we heard about this morning and yesterday about how we're able to use spectroscopy to characterize geometry. Here I'm just going to do conductance measurements. So these are very simple, but hopefully still give us insight into the atomic scale features of our junction. So we're going to use STM break junction technique, where we smash the tip into the substrate, break these contacts in the solution of molecules, and are able to trap a molecule between the tip and the substrate. So these are the kinds of measurements that we measure. As you know, one of the advantages of STM break junctions is that we're able to do this thousands of times and get really good statistics. So I'm going really fast because we've seen this all before. So the molecules we like to use are Amine-Terminated molecules. We find that they give us very narrow conductance histograms. So here the conductance features are really quite a bit smaller than an order of magnitude in width. And we find that that's convenient for getting trends between molecules. So all of these histograms were constructed from about 1,000 or thousands of measurements without any selection. So every single trace that we measured, we included in these histograms. Okay, so conductance scales with molecule length. But let's look at this a little bit more carefully. As Harry mentioned, we can get conductance information, but we also have displacement information here. And so what people typically do is they assign this sharp break when the G0 junction breaks to be the 0 on the displacement axis, and then they can bin conductance and displacement. And so using this kind of two-dimensional histogram, we can see that this C4 diamine, so this is 4 carbons bound by amine linkers at the end, has a conductance of 10 to the minus 3 G0, and it can sustain about 3 angstroms of elongation on average. But this distance here is a relative displacement. It's a relative displacement compared to this sharp rupture at G0. So can we maybe do a little bit better? Can we actually get an actual distance between the two electrodes so that we can correlate conductance to an actual distance between our electrodes? And so what we can do then is if we push, what we thought we could do is if we push the junctions back together, in addition to this conductance signature, perhaps we can also get a distance signature of different molecules. So that's what we're going to do. We're going to stretch our junctions, and then we're going to push them together. And so what this allows us to do is to get both the pull traces. This is a C5 diamine example, and push traces. Someone asked about what happens when we push. Well, the conductance is quite messy when you push, but what this lets us do is it lets us find the point where the metal electrodes touch. And now we can assign to each point on this pull histogram, on this pull trace, both a conductance and a distance measurement. We can do this for now for thousands of molecules, and now we can form slightly different two-dimensional histograms where now we have a conductance, and now on the x-axis we have an actual distance between metal electrodes. So again, not a relative distance, but an actual distance. And if we do this relatively simple exercise on different length molecules, then we see that indeed longer molecules tend to bind at longer gap distances, which makes sense. But there's another thing going on here. So yes, perhaps we can measure the distance between the tips. But there's another thing happening here, which is that it's not binding at one exact distance. And this was already mentioned. So this is an idealized picture that the molecule really binds at the tip. Instead, there's a range of distances. So this has already been explained. Basically, what happens is after you rupture your quantum point contact, you open up a gap. We call it the snapback distance. And if the molecule is longer than the snapback distance, it can bind higher up on the pyramid. And then as you stretch the junction, it can change attachment points. So here's the distribution of snapback distances we measure. Room temperature, it's peaked at about 8 angstroms. And so the C4 molecule is about 0.65 angstroms in length. And so it can bind higher up on the pyramid. And then as we stretch the junction, it changes its attachment point. OK, so if this is really true, then we would predict that our molecules, that if we just look at the yield of our molecular junctions, then the yield should be determined by the snapback distance. In other words, C4 molecule is about 0.65 angstroms, and so only about a quarter of our junctions or so should have a C4 molecule bound, where something that's maybe longer will be bound most of the time, or will appear in almost every single trace. So this is indeed what we measure. So this is a nice check that our junctions make sense, right? We just built this instrument, so this was a nice kind of sanity check. So in black, I'm plotting the molecular yield. So Harry mentioned this in his talk on Tuesday. So the percentage of traces that show a molecule. And as you can see that the molecular yield scales almost exactly as the integral of the snapback distribution that I'm plotting here. So in STM, this is pretty robust. So we don't see variability from experiment to experiment. Instead, the probability of junction formation is dictated really by the molecule length. So this picture seems to hold here. OK, but can we still use this pushback, this distance that we measure here? I apologize, the axis seems to have run away somehow. But again, the 0 here is the 0 distance between the gold electrodes. Can we use this distance for different molecules as a sort of ruler? You might have heard of plasmonic rulers, where you use plasmonic coupling as a distance ruler. Here we're looking to use conductance as a distance ruler. And indeed, you can see that C7 on gold and silver gives you a similar distance, average distance, whereas different length molecules give you different distances. So this is fairly simple, but it's still, again, it's a nice check. So if we plot the mean metal-metal distances we measure using our pushback technique against molecule length, we see that for alkanes, it follows this linear trend with a slope of about 1. So what we think we can do is use conductance of our alkanes. We've heard that alkanes are often the fruit fly of this field. We can use the distances we measure with alkanes as a sort of ruler to check molecules that maybe have multiple conductance features. So phenols in particular, if we measure diaminophenyls, they don't follow this trend. They seem to bind at shorter distances on average. And as I'll show you in a minute, they actually have multiple conductance features that we can now use alkane rulers to understand what is the geometry for oligophenols. But first, let's just quickly look, why does this work at all? So I just told you that molecules can bind higher up on the pyramid, and then when you stretch the junction, They can change attachment points. So for alkanes, why do we measure this almost one-to-one agreement between stretched molecule length and gold-gold distance? We turn to density functional theory calculations to try to understand what's going on. And so we systematically probe the available junction geometries as a function of metal-metal distance. And so if the metals are separated fairly far apart, then the molecule can only bind in this extended configuration in the trans geometry, where it's as extended as possible. Whereas as you bring the junctions closer together, what happens is that there are these cis conformers that are actually lower in energy, but they don't conduct. So these conformers don't have a conductance that we can measure. But if we look at the energy of binding or the overall energy of the junction, then these red ones are the non-conducting cis conformers. And so for alkanes, what we think happens is that the junctions are dominated by these more extended geometries, which is why we predominantly measure a gold-gold distance that scales with molecule length. So again, why is this important? Well, for us, it's a nice benchmark of our instrument, as I already said. But I think it does let us do things that are potentially interesting with this very simple technique. So if we look at oligophenyl's, I've already told you, I've mentioned briefly, that they actually have two conductance configurations. And there's been discussion in the literature about what these are and various hypotheses. But what we can do now is apply our alkane ruler. So the high conductance geometry binds or appears at metal-metal distances that are on average shorter than the metal-metal distance measured for a similar length alkane. So this suggests that this conductance feature is probably corresponds to a molecule that maybe binds higher up on the pyramid. And this makes sense because oligophenyl's don't have cis-trans conformers, right? They can only they're conjugated stiffer molecules, and so it makes sense that this one can bind higher up on the pyramid. This lower conductance plateau, in contrast, binds at longer metal-metal distances. And so what we think is happening is that we have a pi-pi stack geometry. And in fact, there was a paper published in 2018 which hypothesizes that this geometry is responsible for this lower conductance configuration. And our measurements here agree with that interpretation. So what I've told you so far is that we use amine-terminated molecules, and that amines are convenient in a way because the conductance of at least aminoalkanes is not sensitive to geometry. So it forms this donor-acceptor bond to gold, and then as you stretch the junction, even though you change the attachment point, the conductance always stays the same, and you get a very narrow distribution of conductance values, which allows you to compare across molecules. And in fact, this remains true if you go to high vacuum conditions. You still have a very sharp metal peak. The noise you see here is just because we have fewer statistics in these conditions. But junction conductance is not sensitive with pressure. What was really surprising when we did these measurements is that we found that actually it is dependent on temperature. And we saw that as we changed the temperature, the conductance peak shifted. We didn't expect this, and we we were using a low temperature or variable temperature STM at Brookhaven National Labs, and we warmed it up all the way to room temperature just to make sure that the lower conductance we saw at lower temperature wasn't some sort of electronics artifact. So before we go into why this is happening, just a brief overview of how we do measurements at 4 Kelvin using STM-controlled break junction. So we deposit our molecules on gold 111, and then we come in with our tip, and we smash into this pristine, beautiful layer of molecules. And what we actually found is that if we didn't move our tip over between every single measurement, we didn't have a conductance peak. So moving over between each measurement was critical for us to get statistics and get enough measurements to create a peak in our conductance histogram. And of course, the nice thing about using a proper STM is that you can now go back and you can image your surface and you can see what was the effect of smashing into the substrate. And not surprisingly, we saw we formed these huge mountains, which will become important later on. OK, so this is what data at 4 Kelvin looks like. And as we raised the temperature, we saw that the conductance went up. And again, this was statistically significant. We made histograms. And again, we see now statistics are not as good, but there is a clear trend towards higher conductance. So at every single temperature, we got a peak, except at 4 Kelvin, where we seem to get just this ugly tunneling kind of background. So very briefly, I'll tell you the story at 4 Kelvin, because again, it relates to the temperature dependence overall. So we were trying to understand why at 4 Kelvin we don't see nice conductance peaks in our histogram. And so we looked at the snapback distance, right? So the focus of my talk is geometry. So the story here, again, will be geometry. Geometry affects what we see for single molecule conductance. And so at 4 Kelvin, the snapback distribution changes. So the gold is so frozen that when you rupture it, you don't open up a gap. Instead, in many junctions, as soon as you rupture, you actually have zero distance between your electrodes. And so what's happening, we thought, was that in these short snapback junctions, tunneling was dominating conductance through the molecule. In other words, tunneling through the vacuum was dominating. And so if we just take out the traces that have a short snapback distance, we recover a conductance peak. So the solid line is just all the junctions all together, whereas the shaded histogram is what happens if you remove these short snapback junctions. And indeed, you recover a molecular peak because, again, you remove tunneling through the background. So what we see then, to summarize, again, we see that the conductance increases with temperature. And this is also true at 4 Kelvin, except that the tunneling background can mask your molecular signature unless you remove those short snapback traces. OK, so what's going on? Why do we see this monotonic increase in conductance with temperature? This is tunneling transport. We heard a lot about how if you have temperature dependence, then it's hopping. And if there's no temperature dependence, it's tunneling. Temperature doesn't enter anywhere into the equation if you assume tunneling regime. So what's going on? We saw this in 2 molecules, by the way. So, actually, I'm just going to, well, all this shows is that if you take a measured dI/dV curve and you consider Fermi function effects, they cannot explain this temperature dependence. So the smearing here is completely negligible compared to the position of your HOMO relative to Fermi. So we think something else is going on. And basically what we think is going on is the effect of the surface roughness. So at 4 Kelvin, we have a lot of these junctions that have zero snapback. That means they're really corrugated. And so you're binding your molecule at the tip of this tall pyramid where the surface isn't as effective at shielding your charge. And we heard about this, I believe, 2 days ago. And so the effect of this corrugation of the surface is that the effective work function has changed, right? So again, when you're at high temperature, your surface is much more flat. Your molecule is bound on the surface, and the dipole can be screened by the electrons in the metal, whereas when it's bound on the sharp pyramid, that screening isn't as effective. And so the resulting effect is that effectively you shift your Fermi by something like 1 eV. If you plug in all the numbers, including the radius, this R is the effective radius of this protrusion here, you get a shift of about 1 eV. And so if you use the single transport channel that Harry mentioned, and you imagine what happens when you shift your Fermi energy by about 1 eV, you expect a sizable shift in conductance, and we think that's what's going on. I think there are other effects. Probably the density of molecules on the surface also changes, and this can further shift your Fermi function. But so again, we think it's kind of the geometry of the junction that affects the conductance that we see. So tunneling is not actually temperature dependent, but you can get temperature dependent effects because effectively with temperature you're changing your surface, and that can be important. So with this in mind, we have just started measuring conductance of ferrocene at low temperature. I just wanted to very quickly, I think I'm out of time, show this one last slide. Again, we do exactly the same thing. We deposit ferrocene on gold. In this case, we find we really get very, very ugly images. Basically, ferrocene doesn't seem to stick to gold very well, and so as we are imaging it, it's just moving around. But it seems clear that it's a monolayer. So we can, again, do the same experiment. We smash, we pull out. Sometimes we don't catch a molecule, but if we keep repeating this, sometimes we do. And so this is just, OK, this is what we measure. These are, so blue is we pull out and pull in, pull out, and black is the data. And again, this is my last slide, basically. I wanted to show it because it's similar to what we saw yesterday of ferrocene on gold. Sorry, that was ferrocene on silver. This is ferrocene on gold. So again, we get these features at about 0.01 G0. And the vacuum at 4 Kelvin, yeah. And the plateaus here are about half a nanometer, which we think is consistent with kind of the geometry that DFT predicts. So with that, I'd just like to thank you for your attention. I know I was standing between you and wine tasting, so thanks for bearing with me. Of course, I want to thank my collaborators and the students in my lab, and thank you for your attention.
Thank you.